Good morning, friends. Now, this is a series of lecture related to ultraviolet visible spectroscopy chapter. Ultraviolet visible spectroscopy. What is the principle of ultraviolet visible spectroscopy? Here, there are some interaction takes place with of ultraviolet visible radiation with the matter. So, UV visible spectroscopy. In UV visible spectroscopy, UV visible radiations are interacted with the matter. Now, here from the last some few lectures, we have learned about the instrumentation regarding the spectrophotometers. In now today's lecture, we will focus on the cuvette. So this is a part of, we can say, instrumentation, that is cuvette, which is a very important part. Now this cuvette is also called as a cell. Cell is nothing but the cuvette. Okay. Now if you see this diagram, then these are the monochromatic radiations which are filtered here, and the path of this is the path of monochromatic light. Okay. Now this path of the monochromatic light is focused on this radiations. We can say this cuvette or cell. Now this path of the monochromatic light is incident on this cuvette. Now the cuvette contains the solution. It contains the solution. Now after this light will be transmitted after the cuvette. Now the light is transmitted from the cuvette or cell. That means some part of the radiations of monochromatic light is absorbed by the solution present in the cuvette or cell. So this is the most important or crucial part of the we can say any type of photometer. Now if you look the theory, cell or cuvette, it is the solution container used to hold the solution in a path of or in a beam of monochromatic radiation. So it is a solution container basically. Cuvette or cell is also called as a solution container. And what is its role? To hold the solution in a beam of monochromatic radiation. So it holds the solution. It keeps the solution in a beam of monochromatic radiation. It is a simple cylindrical or rectangular glass tube. So if the cylindrical glass tube we use, then cylindrical glass tube is used in colorimeters, and a rectangular glass tube is used in a spectrophotometer. So this is the most important difference. Cylindrical glass tube is used in a colorimeters, while a rectangular glass tube is used in spectrophotometers. Now in colorimeter, cylindrical glass tube is made up of glass material, and in spectrophotometers, rectangular glass tube is made up of quartz tube. Mostly rectangular glass tube of 1 centimeter width are used. So what is the width of the rectangular glass tube or rectangular quartz tube? It is 1 centimeter. When measurement to be done in UV region, then the cell made up of quartz tube is used. So in measurement of spectrophotometers, especially in UV region, then cells of quartz tube, since glass absorbs UV radiation strongly. So this is the limitation of glass tube, or you can say cylindrical glass tube. Glass tube cannot be used in UV region, or you can say especially in spectrophotometer, because there are some problems or error arises because glass absorbs some amount of UV radiation. So it encounters, it opposites, it creates the problems in the measurement in UV spectrophotometer. Now this is the shape of if you focus, if you look this, then rectangular glass tubes. These are the rectangular glass tubes which are employed in UV spectrophotometers, which are made up of quartz. Now this is a cylindrical glass tube. It is made up of glass and it is used in colorimeters. So this is nothing but the 1 centimeter, which is the whole width of the we can say any type of cuvette or cell. This is the width. This is also the width, which is also the 1 centimeter. So this is all about the we can say cuvette or cell in any type of photometry, such as colorimetry and spectrophotometry. With this content, I conclude this lecture. Thank you very much for patience listening. I again request to all my viewers to subscribe my channel, so that yeah, I would emulate. I will be emulate, and I will get a hope to do some part of this video uh, uh, preparation, so that I am interestingly done this job. Uh, with this content, I conclude this lecture. Thank you very much.